It's that wee yellow thing in the corner again, with that little word live written in black. Do I believe it? Well, I think I'm about to find out, because I'm going to do a Jay-Z Kane as usual. Yep. I've done my usual Jay-Z Kane, which means it's working. Good evening, people. Sorry about that. Here we are. Right. Tonight, for one night only, here are the science bitches. Me and Jade, with our collection of honorary bitches around us. They're very nice bitches, aren't they, Jade? Now, we're talking tonight about something that a lot of people think is a blast from the past. Diphtheria. Nasty disease. There used to be a lot of diphtheria around. Today, and not so much. There are reasons for that we will get into. But one of the things is that there are areas where diphtheria is making a comeback. And there are a lot of people that don't know much about diphtheria. It's one of those diseases that everybody's heard of, but they're not always quite sure what it does. They know it makes you pretty sick, but the actual pathology of the disease, what it's actually doing to you, and how it manages to do it, these are things that people aren't aware of. And while they know it is a serious disease, they're maybe not aware of just how serious it can be. And that's usually down to how it produces the diphtheria itself. So this is a brief introduction, and then I'm handing over to the real experts. Diphtheria, it's a bacterium. Corynebacterium diphtheriae. And there are 4 subspecies, but really sort of in my day there was only 3. They had a little suffix at the end of the name. Mitis meaning mild, intermedius meaning middling, and gravis meaning serious. And depending on which subtype you were infected with, it would depend on the seriousness of the disease. Now, this bacterium, it's a funny one. It's a corynebacterium. They're usually found in soils and things like that. And it's, for the actual shape of the bacterium, it's described as pleomorphic. It's rod shaped, but it's swollen at one end sometimes. So they describe it as a club, but it doesn't always have a club shape. And so its morphology is a bit changeable under the microscope. So it's pleomorphic. And it also has intracellular storage granules that you can stain up for. That's diagnostic. When you look at it down the microscope, the rod shapes form what they call Chinese characters. This is supposedly typical. There are all sorts of things about this bacterium. And one of the things it does is, in a low iron environment, is it produces a substance that chelates iron, i.e. it scavenges iron, and it only produces it when there's very little iron around. When there's plenty of iron, it doesn't need to scavenge. That substance is toxic to us. And the very interesting thing about it is that the gene encoding that toxin is not part of the bacterial chromosome. It's a phage, a bacteriophage. And a bacteriophage is a virus that infects a bacterium. And if the Corynebacterium diphtheriae don't have that phage, they can't produce the toxin. So the toxin is phage mediated. It's really interesting, that. They did a lot of work on the phage. I think it was in the was it the late 50s, early 60s maybe? They discovered it was encoded on an extra chromosomal element.
Yep.
A bacteriophage. And it's just, that to me is fascinating. Now, we can look at Why on earth does it have to rely on something that is not part of its own chromosome? But the fact that it has it means it can exist in much lower iron levels now. The problem is that that toxin has side effects on the human body. The biggie being, and we'll get into the details of it, it inhibits protein synthesis. And as we all know, every cell in our body, just to remain alive, has to be synthesizing proteins. If a cell stops synthesizing proteins, it dies. So if you introduce a toxin that stops protein synthesis, the cell will die. So there's one mechanism of action that we're already mentioning, and we'll go into that in much more detail later. Now, what I'm going to do is we're going to cover the icky stuff first, because if any of you out there are like us, we love the icky stuff, and we think you might like the icky stuff too. So I'm now going to hand over to RSC and say, go with the icky stuff, please. Thank you.
Thank you for that wonderful introduction, Fiona. And then, Cliff, if you could pull up the slides I gave you, we can go through them. Hopefully everyone in the chat and everyone who's watching can see the slides.
Yep.
Now, if you were to, okay, great. If you were to make a culture of diphtheria, here is what Fiona was talking about in terms of what the bacteria look like. Now, there are characteristic shapes to bacteria. Usually they're cockeye or circle-shaped or rod-shaped. But this diphtheria has kind of an in-between shape that Fiona was mentioning earlier. Go ahead. Let's go to the next slide. That was a nice little introduction picture. But some basic background. The word diphtheria comes from Greek word leather hide. I'm not entirely sure about the history behind the word, but as Fiona said, it's an aerobic gram-positive rod, which means it can handle oxygen. It needs oxygen to survive. And its main infective particle is this exotoxin we were talking about. The exotoxin is not present in the native, normal, everyday Corynebacterium diphtheriae. It's only present in the ones that have been infected by that phage that Fiona was mentioning. I'm going to let Ian talk about that a little later in depth. So let's go to the next slide, Cliff. So here's what the colonies would look like if you were doing something like a microbiology lab. You would have to do a special swiping or swabbing pattern, get the diphtheria out into little samples where you would actually be able to see individual or paired bacteria. And like Fiona mentioned, the first investigators who saw this bacteria, they thought they looked like Chinese characters. And that's partly how diagnosis was made in the past. When you saw that classic Chinese letter type appearance, you knew you were dealing with diphtheria. All right, next slide, please. So let's talk about how people get diphtheria and where it comes from. So like we said, there are really two types of diphtheria we're talking about today. There's the native strain that does not have the infective particle, does not have the exotoxin. Humans are actually the carriers of this. If you haven't been vaccinated, you could potentially be carrying this bacteria with you everywhere you go. For most people, 90 to 80% of people who carry this bacteria, they never have symptoms. Their body has built up like a balance. And this balance keeps the bacteria in check, but at the same time, the bacteria is still present and still able to grow and reproduce to a limited degree. Transmission comes through close contact. It can also come through respiratory droplets. So people in the hospital are under special airborne precaution, respiratory droplet precaution. So patients and nurses, they are protected using masks and appropriate gear. Gloves.
Well, now, when you say vapor, do you mean like sneezing or sweating?
Sneezing, because it's mainly a disease of the mouth. and esophagus. Not much so through sweat, but you would want to protect from all forms if someone had diphtheria and you were trying to take care of them. And because it's... did you have another question, Cliff?
No, that was it. I just wanted to clarify.
Okay. Well, thank you. Because of the way that it's spread, we see a similar pattern to the influenza spread that's always in winter when people are spending more time indoors in close contact. That's when you'll see spikes. But luckily, for the most part, since the 70s, in the US, there haven't been a lot of outbreaks of diphtheria because of the success of the vaccination programs. If I could give you some numbers, back in the 1920s, diphtheria would affect about 2,000 people per 1 million. And it was very serious. You're talking anywhere from 30% mortality if you got the diphtheria with the infectious exotoxin. But now the vaccine program has done such a tremendous job at reducing those amounts, you only see 0.00001 infections for every million people. So we're talking 10 to the 6th power reduction in the number of cases because of the availability of the vaccine.
Let's just take the opportunity to re-express that number in terms of what it means to a population. So for clarity there, we're reducing it if there were how many cases to begin with?
Approximately 2,000 for every million people in the US.
And now the rate is?
100,000th
right.
of an infection per million people. So we're lucky. I mean, we're not lucky. It's 4 or 5 cases a year would be the normal. And that's usually people traveling and bringing it back with them from somewhere else.
So with 10% mortality, instead of 200 people dying, statistically, no one should.
Correct.
Wonderful. Great. Thanks.
And this disease doesn't spare anybody. Age, gender, socioeconomic status. Everyone is just as likely to be able to pick up this bug, unfortunately. As long as you're not on antibiotics and you're not treated for this infection, you could be carrying, transmitting, or passing this disease on. Just want to take a chance here and see if anyone in the chat had questions or anyone in the Hangout wanted to chime in with their 2 cents.
Well, I could chime in on the communicability issue, and yeah, I'm certainly. So we have the live chat going, and we are monitoring the live chat. So questions and comments, certainly throw them in here. Yeah, it's interesting. So, I mean, as most people realize, and bacteria doesn't do itself any favors by killing off the host, right? So diphtheria in most people lives fairly comfortably, and it tends to really like mucosal tissue, where it lives quite happily, and it's a kind of a nice place for bacteria to live because the occasional coughs and sneeze can aerosolize these small rod-like bacteria, and they can get into other, they can actually survive on surfaces for some time, and we're talking the order of hours, not days. But it makes for, it sort of lives comfortably in its host, and lives in a place where coughs and sneezes can eject to create more hosts. The weird, almost science fiction horror film aspect of it is that there's this bacteriophage. It's basically it's a virus that infects a bacterium that infects humans, turns it into this awful cytotoxic factory. Did I say abacteria? Shame on me. I'm getting the scolding in the side chat. I think I said abacterium. I think first I said bacteria, then the second time I said abacterium. But I don't know. We'll have to go back and listen to it. But it's maybe the third time I said it. It's a virus that affects a bacterium that infects humans, and that system sets up this production of this cytotoxin that very harshly affects, especially like neuronal tissue. So it gets into nerves, and it actually does a lot of damage to nerves, which creates problems for the brain and the heart. But if it just killed the host, well, then there wouldn't be any more hosts. But yeah, it presents pretty unpleasantly when that bacteriophage is present.
It's, if I may, one of those strange ecological balances that is entirely consistent with natural selection. the infection of the bacterium by the virus allows the virus to propagate as the bacterium does better. There's no intent, there's no teleology or anything of that nature involved. This is an organization that is extremely productive for the bugs.
That's right. I didn't see any questions in the live chat. Did anyone see anything?
No, not at the moment.
So
Okay.
maybe could we discuss symptoms?
Yeah, I think actually it's the next slide, if you would.
Okay,
Cliff.
sorry, I'm just excited.
So yeah, let's talk about how this infection develops. Basically, the bacteria is going to be transmitted from someone who's a reservoir, a host, who's not who's not showing signs, most likely, and then it'll be passed to the new patient. And in 2 to 5 days of incubation, the bacteria will be able to multiply and grow to the point where it does start to cause symptoms. And basically, the symptoms you get are based on where the bacteria attacks or attaches. You can get it in the nose, the pharynx, the tonsils. Those are some of the most common places. A little further down in your throat, like the larynx, not as common. Very rarely do you get it in eyes, which is ocular, or in the genital region. But there is another form that is called cutaneous, so the skin. So your two sites for infection are basically the mouth and your esophagus, or the skin. And they look very different. The bacterium acts differently in each location. Anyone want to add anything at this point?
Yeah, is it time for Fiona's hideous photo so that I can get it off my other screen and staring at me?
Yeah, icky picky.
Yeah, let's do it. Jump to the pictures.
Cutaneous form. The icky picky.
I've had this on my other monitor staring at me for about 20 minutes.
It is.
Yeah,
It is.
that
I put
is-
the live stream over the top of it, so so I didn't have to look at it.
That's a cutaneous form of diphtheria, which
Yes.
forms a really nasty little leg ulcer. And that hurts.
Yes. So the cutaneous form, it looks terrible and it feels terrible. There's no sparing of nerve endings. So patients with this kind of problem are going to have the full sensation and the full pain that go along with a wound that looks like that. It will eat through skin, tissue, and if you give it long enough, bone and blood as well. People who are more susceptible to this cutaneous form, people who are homeless, people who don't bathe frequently, alcoholics are especially, for some reason, quite adept at picking this up, especially if they are the kind that are out on the streets. Other signs and symptoms that go along with this go along with almost any kind of infectious process. You'll feel sick, you'll have a fever, you can feel weak. And then, like I said, the symptoms are based on really where it is. So do we have some more pictures of
Do we have a pseudomembranous nasopharynx?
We should, I think, in the next slide or so.
Jump next one.
Okay, well, maybe it's one more, but I mean, this is just basically restating what we said. So yeah, so here's the kind of picture you'd see if you were looking in the mouth of someone who had diphtheria. The exotoxin has certain distinct properties that cause it to build this fibrin sheet. This fibrin sheet sticks to the throat, sticks to the tonsils, sticks to any mucosal moist surface, and once it's stuck there, you can't remove it. If you want to get rid of it, it requires surgery. It has to be cut away. That's one of the things that is a hint that you're dealing with diphtheria. Of course, other problems that go along with having this membrane in your mouth, in your tonsils, would be sore throat, hoarseness, change in voice, and also difficulty swallowing. If you are unfortunate enough to have this infection go further down the esophagus, it can cause difficulty breathing and wheezing, cough as well. Those are the worrisome symptoms, because if the fibrin sheets start to block your airway, they have to stick a tube down your throat, and if they can't stick a tube down your throat, they have to cut your trachea open, put a tube in there to keep you alive.
Nice.
Yeah, actually, I'll chime in on this point a little bit. And people who want to do some research to find more of these photos, a pseudomembrane, if you look for diphtheria and pseudomembrane, you'll see a lot of these photos. Most of these present this way with, oh, well, you know, a tongue depressor. This one's using 2 tongue depressors to show you the pseudomembrane, the fibrous pseudomembrane at the back of the throat. But that's, strangely, these are the rarity. They're more commonly and more aggressively, you see them in the larynx. And as RSC points out, traditionally, I'm talking about the late 19th century, early 20th century, the main route of death, and brace yourself how gruesome this is suffocation. So you've got this infection and it's painful, and eventually the person dies simply because they can't breathe anymore. And how slowly would that form of, I mean, suffocation is a bad enough way to die, but generally when people in circumstances of suffocation, they're suddenly in unexpected circumstances. In this case, this fibrous membrane grows slowly over a period of days, with it becoming steadily more difficult to breathe until eventually the suffocation leads to a fatal hypoxia. So as I pointed out, these ones at the back of the throat are not the more fearsome ones. They're just the ones the easiest to get the photos of. And then the RSC mentions intubation. It's not very often that somebody, it'd be pretty hard to die in a civilized country in 2018 of suffocation because of this exuding laryngeal infection, because they just intubate you. But that's not a comfortable procedure either. So yeah, gruesome.
Thank you, Hogtie.
Just a quick thing. Fiona in the side chat just said, remember, this is a pseudomembrane. It isn't a real membrane. It just behaves like one. What does that mean?
Fiona, do you want to take that?
Yeah, I was just going to say, it isn't actually the way we would normally think of a membrane, but it's like a fibrin clot. You know how fibrin within your plasma, it's got all the various clotting factors. And so you manufacture a sticky clot to seal a wound. Well, this fibrin type clot in the throat is doing the same thing, and it's sticking on tight because that's what it's supposed to do with a wound.
Yeah,
and that's why you cannot get it off.
It's basically forming the beginning of a scab.
Yeah.
Over a trauma site, albeit a small one caused by the bacterial exudated toxin.
And anybody who ever goes into a nice hot bubble bath with a scab on their leg will know that if you stay in long enough, the scab will go white. And that's because scabs are dry things, but in that moist environment, it cannot dry out.
This is the only podcast that you can get scab recognition tips.
Yes. And the ones that you can pick and the ones that you can't. We kept my son's verrucas in a jar for 5 years, for God's sake, in this house. Anyway, yeah, we did. Anyway, sorry, RSC. So it's behaving like a membrane, but it isn't actually a membrane. It doesn't have the structure of a membrane, but it behaves like one. And that's how it's able to actually cut off the airway. And it's that stickiness as well. It means you can't cough it out. It's stuck. Sorry, on you go.
Well, yeah, it's fibrous and integrated into the tissue. Yeah.
Yeah. So, Cliff, did that answer your question?
Yeah.
Okay, great. Let's continue on. I think I only have maybe like 6 more slides left, so let's keep it rolling. And so here we have some more pictures. You can see in the back of the throat more examples of this pseudomembrane. It's a classic finding.
It looks almost like someone's had the teeth punched in and they're still stuck at the back of the throat.
Yeah, and I mean, it's not just you see the pseudomembrane, but you'll see redness and irritation, even bleeding that happens from the infection. So, and here is another characteristic finding, the bull neck. This is a very classic finding that's associated with diphtheria, because that pseudomembrane starts to cause inflammation and swelling in the head and neck area. And when you have this unequal swelling pattern, that the neck pops out real big, it gives you this bullfrog type appearance. And that's where the name bull neck came from.
And I'm going to take Sabella's comment in the live chat as a question. She says, that looks hella painful. It is actually pretty painful. One of the tests of the test on palpations, you want to palpate the lymph nodes, and there's actually resistant pressure, and the patient will often sort of yard back from you because it's actually that level of swelling where you get bull neck is actually quite painful.
Mm-hmm. Very rare nowadays because most patients don't make it that far. They get treated well before the infection gets that bad. So
Speaking as
again,
can I ask, speaking as a clinician, or I suspect too in the panel, is this a thing that trainee doctors and junior physicians are actually seeing in practice?
not here in the U.S. We do have physicians that travel outside of the country to Africa, to Asia, and they will see that there, and they're specifically given refresher courses before they go. And of course, they're also made sure to be immune, that their immune status is protective for them to be going to these places to provide this kind of care. But you're right, because of the statistics I was telling you, that maybe there's 5, maybe there's 10 cases in the year, 99.999% of doctors in this country will not see this infection. But because it's still tested on the board exams and everything, it should be very easy to catch, because everyone is expected that this kind of a condition, it's been so well studied for over 100 years. It's a cornerstone to the history of medicine in this country and in the world. It's just required knowledge. You have to know it. There's no excuse to not know it.
Yeah, ditto that answer for Canada as well. I mean, I've never seen it. I never taught anyone. I mean, there are a lot of conditions that happen around the world where a lot of doctors and students would have never actually seen the condition, and they're shown video clips or slides or textbook images. And this is one of them. I mean, I don't even know anybody who knows somebody who's seen diphtheria until this last year. And but yeah, it is still on the board exams. Federally, it is still listed on the what they call the reportable communicable diseases. So it's as a matter of law, doctors are required to be able to recognize it and then to report it when they see a case of it. So it still appears on all the board exams, and these sorts of images are available. But like my earlier comments about the palpation on the neck, this is based on a review of training documents that go back to the early middle 20th century. Right. I mean, this is just not something people are seeing anymore.
This is a resonant point, I think, of importance for the audience, that in the Western world, we're privileged to never have to deal with this kind of thing. But this is a function of a developed culture and vaccination and other preventative and treatment options. For most of the rest of the world, diseases like this, not necessarily this one, are a day-to-day mortality hazard. And it's something that we need to be focused on, I think, to understand the of epidemiology.
Yeah, sorry, just one more quick thought. I was actually taught it was more likely that I'd encounter a case of schistosomiasis than it would be that I'd ever see a case of diphtheria.
Yeah, probably.
That's an amusing joke for people who know what schistosomiasis is. But anyway,
that's true. I've encountered schistosomiasis.
You have?
4 or 5 times, yes. Well, I mean,
are you kidding me?
You got to work at that.
Let's put a pin in that and come back to that. That is bizarre.
Well, I've been everywhere. I've been out west. I'm in Ohio now, so I mean, I've been all across the country, which is why I've been in the places where it's endemic, and you see it every so often. But anyways, let's get back on topic.
I got a quick question, actually. One of the things that apparently has happened in over the last decade is tuberculosis has sort of come back in the UK, because you've got people traveling from all over the world and things like that, and where they came from. It wasn't, it's not a standard vaccination most people get or whatever. Would diphtheria be the same, that people could bring it in? Is it quick to diagnose? Is it quickly say, oh yeah, know exactly what that is? Or because it's such a rare thing for Western doctors, is this something they would think, I don't know what that is, let me just check?
Well, the thing about it is, like a number of other bacterial diseases, you can carry a smoldering, self-replicating infectious dose around with you, and it doesn't cause you any disease. Some would say organisms like this are commensal in that they interact and don't cause pathogenesis most of the time. And there's a trigger factor that results in a pathogenic response. So just like you could swab people's noses and find Neisseria meningitidis, they're not having any kind of syndrome.
Okay.
Right.
Yeah, and actually, Cliff, we'll come back to that one in my segment. Ask what happens when doctors who've never actually seen diphtheria, what happens when they encounter diphtheria for the first time in a world where they think diphtheria was eradicated. We'll come back to that on my segment.
Yeah,
I'm looking forward to hearing about that, Hogtie. But I think Ian made a great point. We are very privileged to be in a society where this isn't really a major issue, medically speaking. But I feel very confident that the networks we have here in the U.S., the lab technicians, the medicines you have, if someone wants to come in with this condition, even if the doctors made a mistake and didn't catch the symptoms and match it to the correct disease, the antibiotics that they're going to give them is going to cover this infection. And B, when they send the swabs in the throat or the swab from the wound, they're going to grow that organism. They're going to see the little pleomorphic comma shape. And because everyone has it ground into their head, that diphtheria has this classical look in the microscope, I think it would just flow from there, that it would be picked up by someone along the line, because there's just so many people that information would have to pass by. And it would be very difficult for that many people to make the same mistake. But that was basically the end of my clinical talk. Here's where I had a couple images describing how the diphtheria toxin causes.
I'm sorry, before you get to that, because we want to talk about the treatment of the antitoxin. But one of the things that I find sad about when you're showing some of these severe things that happen, people should remember too that it's not a terribly egregious infection too. It's a gram-positive infection, and I want RSC to correct me what the modern protocol is. But I mean, things like erythromycin. I mean, they're very. It's a bacterium that's fairly easy to kill once you identify it. So in cases where it sort of gets away on somebody, I mean, it's something that should be easily treated once you encounter proper health care. So I know you want to mention like how it's dealt with in the event that somebody has seen a case. And then, yes, we need to talk about toxin and antitoxin, because we get into the great science fiction horror movie aspect of why this thing is so bad for your nerve cells when it infects your throat.
Yeah, you're right. Gram-positive bacteria, anyone who comes into the hospital with an infection that would make you feel sick like that is going to get antibiotics that attack gram-positive bacteria. Because of the way that the antibiotics behave, they're going to be able to manage a diphtheria infection. However, the optimum medications that have been studied, penicillin still continues to be the #1 medication choice for this illness.
Can I comment?
The speed at which penicillin actually knocks out the bacterial replication and leads to the bacteria dying and clearing of the infection is actually quicker than with erythromycin, which I guess was considered the standard in the 90s because it actually prevents the bacteria from making the toxin. So those are the two very well-studied antibiotics that would be used specifically once you know that it's diphtheria, for sure.
As a side note, as a little bit of background reading today, I looked at some antibiotic resistance data from, I think, a Thai outbreak where penicillin was, or there was resistance to penicillin in 1 in 4 of the isolates. So they were recommending rifampicin.
Rifampicin, okay. Interesting choice.
Yeah,
but,
I mean, it's like
when it comes down to when it comes down to resistance, you have to go with what work for that patient. When they culture bacteria in the labs in the hospitals, they will test many different antibiotics to see which one is most effective for that strain. I was just speaking in general. You're going to see on a lab report that penicillin and erythromycin will be the antibiotics tested, and you'll see that they are usually very effective. And you'll choose one of those two medicines based on patient allergies and all the information you have, together. You're not just basing it on one data point alone. You have to take everything together before making a decision what medicine a patient needs. And so at this point, I think it might be time to hand it over to Ian. And Ian was going to talk about the exotoxin. Does anyone else have any questions in the chat, or does anyone in the Hangout want to add anything before I hand it off to Ian?
Well, I just wanted to do a wee linky between you and Ian, because you've discussed these primary symptoms of diphtheria, but we've brought up the toxin a number of times.
Mm-hmm.
So we've looked at these primary symptoms, that they are local symptoms, they're local to the infection. Because the strains carrying the bacteriophage can produce a toxin, we've also then got to look at what this toxin can do. Now remember, the bacterium isn't producing it in order to make you sick. It's producing it because it's scavenging for iron. But it just so happens that because of the chemical structure of the toxin, it is toxic to us. And Ian's going to cover the mode of action of the toxin, and then the symptoms that the toxin is going to cause. Take it away.
Yeah, I'm really excited to do this, and thanks to everyone. Fiona, this is very good fun, and the conversation so far has been really fascinating. So I'm going to go over some of the molecular aspects and tie that into the symptoms, the presentation of the disease that's been put on before, and then talk a little bit about the immunological treatments, specifically the presently available vaccines and the anti-serum, which is an alternative approach, one that's historically one of the earliest interventions in biotech. So it's a very interesting story. And I've got some basically just pictures to prompt some comments from myself, but I invite everyone on the panel and the audience to chime in, because I think there'll be an interesting discussion to be had. So Cliff, if you could cue it up. Thank you. I like the history of medicine and how it affects people. And this is one of the 1950s British, so National Health Service posters about the condition. And quite frankly, it's the summary of why we do what we do. This is a deadly thing that makes kids and others miserable and sometimes dead. And there is a way to avoid it very effectively, which we've discussed in terms of the statistics.
I have a feeling this picture may be a picture of Sarah Huckabee Sanders when she was a child, because I look at that right. Sorry.
It's a very cute little baby. I have a small collection of this kind of poster, and some really bizarre ones come up from time to time. Next slide, Cliff. So we chose exactly the same picture, RSC, because this is a beautiful one. I won't repeat the bacterial morphology discussion. We've had that, and it's been excellent. Next, Cliff. So it's a gram-positive, and that's a term that many people won't really resonate with. What it means is the outer layer of the bacterium is formed from this big polymer called peptidoglycan, which means it's a tough little bugger. It has a waxy or somewhat waxy coat decorated with lipids and lipoteichoic acid. The inner membrane is phospholipid bilayer with proteins through it, and the peptidoglycan is a semi-permeable membrane. It allows access to that but protects the bug. Gram-negatives have a different outer physiology. Next slide. And here, I think this is an amazing picture. This is
aliens landing on a planet.
Yeah, this is thin layer electron microscopy, and these are the bacteriophages that we talked about landing on the surface of a bacterium, and you can see various stages there of ones actually injecting their DNA into the bacterium, and you can actually see it in the three at the top going in.
These lines here,
Yep, that's correct. It's probably it's definitely artificially colored, but it gives you exactly what's going on. These little viruses, these bacteriophage, land on the bug as if it was a meteor and inject their genetic material into it, therefore kidnapping the bug into making copies of their own genome. So the bug will then sprout off and send out phages under certain circumstances. So this is how the base bacterium becomes modified with the toxin that changes it from being a commensal organism not doing a great deal over a long period of time to an extremely toxic pathogen that can cause, as we discussed, death in between 1 in 10 and 1 in 20 people. So,
like I said, science fiction horror movie.
Yeah, it's absolutely amazing. And as a gentle poke at true empiricism, doesn't this look nifty and give the appearance of design? Because it looks like a lunar lander or one of the thumpers from the movie Dune, but it's a very well characterized.
Sorry.
It's very well characterized at the molecular level, each structure here and how they go together and what they do. So the capsid head contains the genome. It's injected through the collar sheath base plate into a specific bacterial host. And we use this technology to do a thing called phage display, where you can actually monkey with the phage genome and get the bacterium, and get progeny phage decorated with a particular peptide. So this is a tool that's used, and it's a very well understood system. Next
question for you. The previous picture where they're landing and injecting, what kind of time frame would it take to do that? I mean, you know, is this happening in incredibly quick time, or is it just like a
seconds?
Right.
Yeah. I mean, it's not something I've physically timed myself, but at that kind of scale, we're not talking about an extended process. I'm sure it has been done. So don't quote me, but it's almost certainly a very rapid process.
Well, if you look at, you know how when you do your bacterial lawns, infecting it with phage and all the rest of it, and you're looking for plaque formation?
Yeah.
And especially when you're using E. coli and you're getting your plaques formed with an overnight incubation. And to form that plaque, obviously, millions of bacterial cells will have had to have been infected and lysed by the bacteriophage just on that overnight. So yes, it can be very, very fast.
So yeah, it's science fiction, and I guess one of the reasons I'm into this is because it mirrors an awful lot of the stuff I was fascinated by in fiction. But it never gets weird enough for biology, and nature will find a way to reproduce its genes. And sometimes that gives the appearance of design. So here is an epithelial surface.
It looks like a sea anemone.
Yeah, exactly. This goes back to what Hogtie was saying earlier on. This is a really nice environment for a bug. It's nice and warm. There's lots of oxygen going. There's movement. So, you know, there's potential for spread. And it's a little bit like losing a raisin in your furry bathroom rug. There are a lot of ways for stuff to stick there. Next slide. And the way the toxin works, once it's expressed on the epithelial surface, is it's small enough to be taken up into cells, and we'll talk about that in a little bit more detail. But the specific receptor for it is the EGFR, which is a growth factor receptor. And this slide shows where that receptor is. The top one, in case people are interested in detail, is a Western blot. So that's detecting the protein in itself. And the beta-actin is a loading control to make sure that the samples are comparable. But what you can see there is this receptor, the one that is recognized by the bacterial toxin, is essentially everywhere important. The last one, it's also a tumor marker for a particular type of cancer, but that's less relevant to this discussion. The receptor for the toxins, liver, heart, kidney, stomach, lung, it's also on many of your immune cells. So this stuff, if it gets in touch with that tissue, can turn off protein manufacturing in any of those organs, and as Hogtie pointed out, critically, neural tissue. So this is a toxin that will basically kill anything it touches if it's present in sufficient amounts. The bottom is RT-PCR, which looks at the level of gene expression, and it just confirms what you're seeing in the top. And the reference is below for anyone interested in this. Next.
Oh, before you go too far, we did have a question from Brian Stevens in the chat. I don't know if you see it here. Ian?
I'm not following the
he's
chat.
Asking, what is the chemical makeup of the collar section of the bacteriophage?
Chemical makeup, it's a proteinaceous compound, proteinaceous structure. I don't have the details handy, but I can certainly provide them.
Okay. Thank you, Ian.
Yep. Pleasure.
Ryan, I hope that was your question.
Yeah, feel free, post questions in the comments, and I'll happily get back to anything. Moving on, one of the reasons this bug interests me is because two of my research fields are related. One concerns iron scavenging by the Neisseria protein, the Neisseria bacterium. Their adhesive properties and their immune signaling for endocytosis and for immunoregulation. So diphtheria does very similar things. But the toxin binds to this particular receptor. The receptor is then phosphorylated and forms a clathrin-coated pit, which is this bubble that forms into the cell. It's then pinched off by the action of various enzymes, including PI3 kinase. And the stuff from outside sticking to the receptor goes into this bubble environment. Then there's an influx of hydrogen ions, which acidifies this internal environment and releases and can actually physically alter proteins that are present within it. And that's how bugs take stuff in to eat, to destroy, or in this case, to poison themselves. Next picture, please. So this, I think this is exactly the same picture as RSC had. So please get involved here. Just to talk it through, the diphtheria bug makes a protein that has two different functions. The first, the B unit, binds to the EGF receptor, which is, as we said, everywhere. And this is a protein that's small enough to get just about anywhere. It's much less restricted in terms of its presence than an intact bacterium or colony. The vesicle begins to form around multiple signaling molecules. And as a function of the acidification, the toxin breaks into two different parts. The A part penetrating into the cytosol, and that's where the business begins. And essentially what this protein does is modifies one unit of the protein manufacturing enzymes, or the enzyme cascade involved in that. And as a function of that, stops de novo protein expression by the cell. And the upshot of that is dead cell. Next slide, please. Oh, I've got the reference on that. It's a very good review of the toxin activity there. So this is
there.
Yeah, this stuff is very well studied, and that's an extremely accessible resource. So that's how the bug does what it does. It does that to, well, the function of it doing that is to have access to more cellular nutrients and to do better, to make more copies of itself and more copies of its bacterial parasite or partner. this is an illness that's interesting in a number of other ways. One, because one of the earliest biotech products was a passive immunization against the toxin itself, because the toxin has been known and it's been able to be purified for decades. Much better now than then, but it was still effective. And a passive immunization is when you're not given a vaccine, but you're given an antibody dose that responds specifically to part of a pathogen. And in this case, the serum was raised in horses. The horses were bled, the antibodies were pulled out using a chromatographic system, now using protein A. So you're basically taking all of the immunoglobulin out of the serum, formulating it and injecting it into people, where it reacts with any toxin present in the blood, and that is then neutralized and cleared. Injecting serum from one animal into another isn't really a great idea, but this is actually very well tolerated and was an extremely effective treatment early on. It's seldom, well, it's, I would say, infrequently used these days, RSC.
It's mainly been replaced because people can have bad reactions to any type of serum. There's always the chance for anaphylaxis, where you get the very bad symptoms, low blood pressure, inability to breathe, things like that. So because of there is a risk associated with the serum. It's greater than 0, but it's not very big. They've gone away from it.
Yeah, exactly. Oh, Hogtie, carry on.
Yeah, can we clear up one little point here? We're talking about, I love this picture too of this horse. Step 2, insert into horse. And this horse is there bridled and with the reins. That's not how it looks when they do this.
No.
So we're not discussing, as Crow Many Clouds asked in the live chat there, do we still use horse serum vaccines? No. We should talk a bit about why the horses were involved here, and I actually appreciate Ian, you were mentioning about this tolerating this serum from other species. So I hope you continue with that point. But I want to make it clear, this is not a vaccine we're talking about here. This is the antitoxin, right?
Yeah, that's absolutely right. I probably clouded that issue by calling it a passive immunization.
Can someone just explain? Can you explain what an antitoxin is compared to a vaccine?
Sure. Right. Yeah. This one.
Yeah. What you're doing is borrowing the immune response from another animal. By dosing the animal with something it reacts to, the animal will make an antibody response to that. Then those reactive proteins can be pulled out and administered as a medicine, where they'd stick to the toxin and take it out of your circulation ultimately.
And is that where you were saying it was a bit of a nasty bugger because it was it had a waxy exterior? Does that make it harder to cleanse?
Okay, no, this is the problem, Cliff. Let me say it this way. So if you come into a clinic and you got those plaques on the pseudomembrane on your throat, and you're having all the symptoms, and you're like, this guy's got diphtheria, there are going to be 2 things your doctor and you, of course, should be very concerned about. There are 2 things in your body that you do not want to have in your body. One is the bacterium that we've been discussing, and the second thing is the toxin that's produced by those bacterium that are infected by this bacteriophage. Right. So the bacteria is in your throat or in your skin or in your nasal passages, wherever it is. But the toxin gets into the bloodstream and circulates around, and then enters into cells and prevents those cells from producing proteins. So your clinical approach is going to be, gee, I kind of want to get rid of these bacteria, this bacterium, because it's an unpleasant agent. But that's easily done with these gram-positive antibiotics. But the other issue is this patient still has these toxins floating around in their blood, and the whole time that it's taking for you to treat this disease, these toxins are still floating around. If you could encourage the patient's body to identify and trap those toxin molecules that are floating around in the blood, you may actually take some great steps towards saving the patient's nervous system. Does that distinction help? And then Ian should explain why the horse is involved. The horse doesn't help you get rid of the bacteria. The bacterium.
I think I'm getting it right now. So the idea is that the antitoxin basically is kind of a replacement for your own, because your own isn't working good enough.
or it's not ready to work. It's like, here's an instant broad immune response.
Oh, of course, it hasn't learned what the infection is yet.
Yeah.
Unless,
And remember,
unless being
something,
vaccinated.
yeah, with something like this toxin, which is one of the most potent toxins known, you can't hang around for 2 weeks waiting for the person's immune system to kick in
Gotcha.
with antibodies to the toxin. So you provide them with antibodies from a different source.
Yeah. And
Ian, do you mind if I jump in for a second whenever you come to a logical spot? Cliff, would you mind going back one slide to show how the toxin goes into the body? I just wanted to clear up a couple of points because everyone has kind of going in different areas. So in this picture, we see a very nice representation of how the toxin attaches to the receptor on the outside of the cell and then becomes engulfed and then gets inside the cell and is able to go and bind to this EF2 factor. I don't know if we mentioned it's called elongation 2 factor, but it's a part of the protein building process for the cell. The exotoxin binds in a manner that's called irreversible. This means once it attaches to the EF2, it cannot be removed. The only way to get rid of it is to completely destroy the EF2 and the whole protein building process and start over and build more of those protein forming factories. So because this toxin binds irreversibly, it makes it very potent, and it is going to damage the cells that make protein all the time. So if you start thinking what cells in your body are very active, well, your brain cells are always making new neurotransmitters and making different assembly parts to keep your brain working. Your heart is always beating. And so that's why the areas the toxin attacks that are most severe are the heart and the brain, the nerves in your body. A few more details about this toxin. The lethal dose. So at LD50 in biology, this is the dose that would kill about half the people exposed to it. 100 nanograms for every kilogram you weigh is the LD50 for this. That is very toxic. I mean, you're talking about less than a fraction of a gram is going to kill a person. So you don't have time to wait, like Hogtie said. You have to neutralize the toxin. It's not just enough to neutralize the bacteria. The toxin is still there.
Right.
So the mechanism that was developed in the 20s before they had all this great technology to be able to see what we're seeing now is they used other animals that were able to build the toxin or build up antibodies to the toxin. They were able to store those antibodies for people who got sick. So the horse serum that Ian is talking about, its purpose isn't to eliminate the bacteria. Its purpose is to eliminate the toxin that's in the bloodstream. And here's the important point, because I said it binds irreversibly. You can't take it out once it binds in the cell. It's only going to take out the toxin that's in the bloodstream that isn't already inside the cell. And that was kind of the point I wanted to make. Does it really give you a distinction between what each thing is doing? The antibiotics are trying to kill the bacteria and prevent them from making more toxin. The serum is trying to stop the toxin that could potentially go and continue to attack heart, brain, or any other part of the body.
Now, how would a vaccination which would already set up the I forgot what it is now, but yeah, the antibodies. It would basically trigger antibodies already.
I have that in the next few slides, and that was a really valuable clarification.
Yeah, and one way to look at it is like, remember the antibodies. Maybe we should mention the role of antibodies. They almost mechanically interfere with things that get into your body. They find something that when they identify something that's foreign, they pile on. Right, like you picture a football game where there's a big pile on, and they completely interfere with this thing. In the case, again, it's not the bacteria that we're talking about. It's this toxin that's floating around. And one way to look at it is, I mean, Cliff, if I gave you an injection of this toxin, the toxin is produced by the bacteriophage infected bacterium, and I happen to have some of this toxin. You don't even have the bacterium in your body, but I inject this toxin into you. It's gonna mess up your brain. It's gonna mess up your heart. It's gonna be really bad for you. But let's say, like 2 minutes after I injected this toxin into you, before it starts to get uptake into the cells, RSC gives you an injection of a whole bunch of antibodies that were produced in some other animal, some other human being, even a horse. Yep. Once they're in your bloodstream, they're not going to care that, oh, I'm not in a horse anymore. They're going to seek out they're already going to be designed to match, identify, and attach onto the toxins that evil Hogtie injected you with. And as long as they bind to them while they're still in the blood, then that would prevent those things from using that mechanism to sneak in through the front door of your neuronal cells, and that injection could, from the horse, from RC, would quite likely save your life.
Yeah,
and this is a particularly cool thing about the animal approach. This is generating what's called a polyclonal serum, which means there are multiple different antibodies that stick to specific parts of the toxin protein, meaning that, as Hogtie says, once it's in there, if these antibodies are present, the whole thing gets decorated all over like a big foosball.
question for you: why would you use a horse and just not take some blood off a human and do it?
Horses are big, and
they produce a lot of blood. You can generate it, take a lot of quantity of blood out of them. And I hate to say this, Cliff, you also don't mind if you kill them in the process. This is part of how it's done. And sorry, one point I want to clarify too about the foosball thing. So you imagine these antibodies are all attached on there. Well, now you've got this toxin that's got, so that here you are, Cliff Jumper. You've got this foreign toxin that I collected from this bacteria that's infected by a bacteriophage, and it's in your body. You don't want that. That's not what you want in your body. And then you've got the horse antibodies that RSC injected into you, and they've all agglutinated together into this big furry ball. Right? You don't want any of those things in your body. Your own cells then would recognize this foreign big blob, and cells, for example, the white blood cells, would then just go and invaginate around these things and actually take them out of your bloodstream. Which is why I was saying it's a way of using horse antibodies as a way of encouraging your own body into helping clean these things out of the body.
Yeah.
Gotcha. Okay.
Although,
by the way, thanks, RSC and Hogtie, you're an arsehole. Don't do that again to me.
And I mean,
if I could just clear up one
RSC,
point about the horse serum. We use horse serum instead of people serum because we found out that people serum has a lot of other really bad things, like hepatitis, like HIV, like immune reactions. If you get like antibodies against your blood cells, you can get a hemolytic anemia. So when they were trying to build serums in the past, they did try using human serum at one point, but they found that people received the serum had such bad reactions so frequently that it became more available to do larger animals that you could mass produce the serum with, like horses, like cows. Horses just were the ones that they found a very quick solution with, and that's why they used horses. Yeah. But Ian, I was wondering, Drag is asking if we could clear up the difference between exotoxin and endotoxin for everyone in the live chat.
Sorry.
Do you want to handle that?
Yeah, absolutely. The exotoxin is a secreted toxin like the diphtheria one that we're discussing. Endotoxin is most often used to describe the outer component, one of the toxic components of gram-negative bacteria. So lipopolysaccharide, which has a ketodeoxyoxylenate component, a lipid component, and sugars. So it's what causes endotoxic shock in gram-negative infections.
Yeah, the endotoxin is basically spilled out when you start attacking the bacteria. So the problem is when people have gram-negative infections in the blood, well, once you start giving them antibiotics to take care of the bacteria, now the toxin, the endotoxin that's in their cell wall, is exposed to the blood, and you get further worsening of the illness because of that.
Yeah, disseminated intravascular coagulation and terrible things.
Correct.
Before we move on to vaccines, which I think is the really cool bit, we don't use a lot of animal products these days. And in my own work, one of the things we do is generate specific B cell lines. And a B cell secretes a specific antibody. And you can engineer those antibodies to improve on what nature does, and then just grow up enormous vats of these clonal cells and purify not a polyclonal serum, but a monoclonal serum with very specific content. It's very expensive to do, but it's an elegant way of modifying the immune response to deal with specific things, not only as well as nature does, but by understanding how it's happening through the protein-protein interactions to actually improve on it. So it's a very interesting way to make a living. Next slide. So this is an example of several diphtheria combination vaccines. And as you can see, this is diphtheria and tetanus toxoids plus the acellular pertussis. I'm going to focus on the word toxoid for this. We've discussed the toxin and how it works. A toxoid is a chemically, in this case, a chemically modified version of the same toxin. So it's the same protein treated, in this instance, with formaldehyde and or glutaraldehyde. What that does is changes the function, or abolishes the toxic function of the protein. It changes that, but does not change the antigenic content sufficiently. And what that means is your body can react to it and make an immune response to a thing that looks almost exactly like the toxin, but which does not have the toxic properties. So instead of injecting a toxin and getting your horse to make the immune system respond, your body is now primed. It knows what to look for. And as soon as that toxin hits your blood, you get clonal B cell expansion. You get a lot of antibody generated very, very quickly to soak up the toxin and prevent it doing the bad stuff we've talked about. If there are any questions in the chat about vaccine contents, we can do that or pin that on at the end with the anti-vaccine component of the discussion. And as a personal note, this is made by Sanofi Pasteur. My first office with Sanofi was actually in the stable where the poor horses used to live. Next slide, Cliff. So there are a number of different versions of this from a number of different companies, and some contain thimerosal, most contain polysorbate 80 as a surfactant. And
what is a surfactant?
It's basically a soapy substance that keeps things nicely in solution, and there's a very, very vanishingly small amount in there. The other contents that people freak out about typically are the formaldehyde and glutaraldehyde. Again, very low concentrations of those. And one that Tommy is freaking out about is aluminum hydroxide, which is used as an adjuvant, which is like a mild irritant that stimulates a much stronger immune response. So that's aluminum hydroxide in this case. Next.
Drag has a question, by the way. The chat, Drag asked, was there a mention of what the immune system is looking for in the non-virulent version, which I assume he's talking about the vaccine?
Yeah, the toxoid represents most of the antigens that the toxin presents. And I'm sure it's been mapped. I don't have the data available, but sufficient antigenic content is present so that there's a cross-reactivity. Your body reacts to the toxoid, and that reaction also works with the toxin. And as Hogtai says, these are big, sticky, Y-shaped proteins that bind onto stuff and basically cover it up and then trigger other immune cells to eat that big blob of foreign material. Another interesting thing about the diphtheria toxoid is because this has been so well studied down to the angstrom level in the various proteins, and the specificities being studied. The toxoid is actually used as a hapten, as a potentiating agent for other vaccines like this one. This is the meningococcal ACY&W135, so it's a quadrivalent meningococcal Neisseria meningitidis vaccine. It's made out of polysaccharide, and it doesn't work particularly well in the absence of this hapten molecule, which acts to stimulate the immune response against, again, antigens from another bacterial species. So we've borrowed what diphtheria does in order to make other preventative treatments more effective. And the meningococcus and gonococcus are very similar to the diphtheria bacterium, in that they cause commensal infections that sometimes end up turning into lethal sepsis. They don't have a toxigenic quality, but they do have a similar immune signaling function, which is something that I've looked into the past. Okay, that's all my stuff. Thank you, everyone, for your patience and contributions.
If I can say something about the toxoid. I want to use the cliff jumper, evil hogtie mad scientist injecting cliff jumper with poisons example again. I'd used the example before. If I had some of that toxin that's produced by the bacteriophage infected diphtheria, then I would, and I inject that in the cliff jumper, would not take very much at all to do significant damage to the heart and lungs and likely cause death, even though the bacterium isn't present. So you wouldn't get the pseudomembrane on your throat or anything. This thing would just do a lot of damage to your body. Now use the example of, suppose I gave you that and then RSC gave you a bunch of antibodies that were produced in a horse. It would actually be able to suck up, the horse antibodies would then attach an agglutinate onto that toxin and would likely save Cliff Jumper's life. If Cliff Jumper, if you were slightly smarter and you were concerned that an evil hogtie was going to inject you with this toxin, one thing you could do is go to Ian first, a year before bumping into evil hogtie, and get the toxoid injected into your body, which has the same the same recognition, the same binding sites, but isn't toxic to your cells. And your own body would learn to recognize it, and you would produce your own human, Cliff Jumper produced antibodies that would be able to recognize the toxin, so that a year later, when you encounter evil hogtie, and I inject you with this toxin, your own body would be able to agglutinate it with your own antibodies. Or you know what? It might also be good if you end up getting diphtheria.
And another little angle there, when you do that, your body remembers. Whereas you get a dose of horse antigen, horse antibody, that will work in an acute sense, but that's cleared from your body very quickly, and you don't have an immune memory because it's not coming from your own immune system. And a thing to point out is the diphtheria vaccines are often administered with the tetanus vaccine that people seem very comfortable with. And people very rarely actually develop tetanus these days. It's exactly the same principle. A toxin treated with a chemical to make it dead, to make it not active, but still looks a lot like the toxin as your body would see it.
I'd like to point out that the way you can tell evil hogtie is his G+ posts are short and have a good amount of brevity in them. Which now means that hogtie can't write a single post that, less than 1,000 words.
To comment for our reality-denying associates, and I won't mention names here because I don't want to engage them, this stuff is so nifty and cool. It really does look designed, but the thing is, it's also been studied way down to the molecular level, to the DNA level, and we're not guessing in the things that we're seeing. And I wanted to mention that the EGF receptor that this toxin sticks to has been mapped in the human genome to chromosome 5, and it's also been sequenced in, I think, more than 50 species. And the evolution of the receptor types and its specificity has been mapped out over multiple genera. So from the echinoderms up, and the evolutionary development of it is also very well understood. So again, we're not guessing about this stuff.
Do we want to? Yeah, I mean, an antigen is. I mean, I don't know if there's actually a specific classification for it. I think an antigen is just anything that would trigger this sort of targeting mechanism by antibodies. Anything that gets into your body, to which your body would then recognize as foreign, or for some reason produce antibodies that in the future, when this thing is present and those antibodies are present, the antibodies, which is the Y-shaped proteins, they would just latch onto the thing, and they pile on by the thousands, like tens, hundreds of thousands, and they just make this big furry ball. So the antigen is basically, I don't think an antigen is like a specific, geez, someone help me out here. Is an antigen a specific class of chemicals? I think it's just anything that anything that would start to trigger an antibody.
Anything that tweaks your immune response, so it could be chemical or otherwise.
Yeah, anything that elicits, this sounds stupid, an antigenic response, i.e. elicits an immune response.
So,
so this could include anything that...
I mean, it's just the target that's recognized by various aspects of your immune system, because this kind of stuff will tweak multiple different systems simultaneously, and they all interact. It's complex and beautiful.
Right. It's like they've been saying, antigen could be any form of protein, sugar, or saccharide, polysaccharide. It can even be a lipid in some special cases. Yep. But the criteria is that it causes immune cells to start producing their defenses.
Anything that sets the alarm off, any breach of the perimeter, will
that's
set
a
off
good way
response.
to describe it.
So you've got something that's not you, and your body goes, this might kill us. It's certainly not us, and it shouldn't be here. Lock it up.
Oh, I think I see why this question is relevant now. The light bulb's going on for me here. The idea of antigenic equivalence. So you have this. So Evil Hogtie's got the toxin, but Ian has the toxoid, where the toxin, if I inject it into somebody, it potentially would kill them, even in fairly low doses. Ian's toxoid would not kill them. It's not harmful in that way. So they're obviously very, very different chemicals. But to say that they're antigenetically equivalent is to say that they would produce the same immune system response. The antibodies recognize the toxoid as they recognize the toxin. So the presence of either in your body would trigger the production of those same antibodies, and those same antibodies would agglutinate onto either a toxoid molecule or a toxin molecule, should you encounter one by engagement with Evil Hogtie or with a bad case of diphtheria.
Yeah, exactly. It's like showing your body's army a picture of the enemy and teaching them this is what you go for, so that when the enemy appears, your body army is ready to attack it.
It's primed.
Yeah, and your body remembers it, which is a really, really cool thing. Although and then it's required that there are a number of repeat doses, and that's a really significant aspect, because while people may get one dose, oftentimes I think the number suggests there's a problem with repetition. So that was all the molecular stuff that I have. It's a very fascinating story, and the couple of references that I put in are really accessible and available online. But I think we're going to move on to Hogtie stuff now.
I would happily do so, and I wonder if that means, I think, is this sort of the last kind of formal segment. So again, I want to encourage people in the live chat to be pretty vigorous with any questions or comments that you have about it. And I'm going to do here is I have rather hurriedly assembled some what do you call it? Chrome tabs, right? So I'll put myself on present here. Actually, before I do that, let me say that one of the funny things that happened to kind of get me involved in this thing was, not many people who are listening to this would know this, but just you'll recognize this. A few weeks ago, I had a bit of a blow up in a hangout because I was very upset with there being a case of diphtheria that had come about in Canada, and actually it was in Alberta, and I was concerned. Because my understanding was we'd pretty much eradicated diphtheria here in Canada, and I kind of had blown up about that, and it was frustrating that, diphtheria is in Canada, like what's going on? And I'd heard a solo hangout that Dragnut had done a few days ago, where he said something about this particular thing coming up, and he said, "Oh, apparently Hawkeye's going to talk about what's going on in Canada, where apparently there's an outbreak." Want to be super clear, there's not an outbreak. I was upset because what I'm going to do, I'm going to go over some of the history of this here, and you'll see kind of why I was miffed about it. But I do want to make it clear to people that when this had happened, I had been in contact with some of my old contacts in the federal ministry and the provincial ministry, and since then I've been in contact with them once again. I've been encouraged to make it clear that it's not an out of control situation in Canada. We had one isolated case in Edmonton, and happy faces. But let's go over where that happy face comes from. Which one here? So this is, I'm going to talk a bit about the Canada's history with diphtheria through the 20th century, the Americans' history with diphtheria in the 20th century, and the world's history. So starting with the Canadian numbers, this is kind of an interesting graph. I wonder, Control plus. So on the y-axis here, we have number of cases, and remember. And then at the bottom, it goes from 1924 up to 2012. Now, remember, this is raw number of cases, not a percentage. So in this period of time, the population of Canada increases about fourfold. So there's more and more and more people in Canada, both through breeding and through immigration, and yet fewer and fewer. You see this line dropping off sharply, and it drops off so sharply that by the time you get to the 1950s, you get this jiggity little bump here. You can't even, because of the scaling of the graph, there's so few cases that they actually had to put an inset graph in here with a different scaling on the y-axis so that after 1955 you could get some sensible information. But even in that graph, post 1980, it goes down so low. Here's the line for 50. We're down here to single digit cases by the time we get through the 1990s. So this graph starts in 1924 because in 1924, diphtheria became what they call a reportable communicable disease. In Canadian law, which means that any physician that encounters a case of diphtheria has a legal obligation to make a report to the ministry that they've engaged this case. Just one moment. One moment.
Hawkeye's having a JD crisis.
Yeah, no, it's all right. They're having fun. And so, yeah, in 1924, it became reportable. So every physician had to report this. And 1926, the vaccine was introduced. So you don't really get what had happened in the early part of the 20th century and the late part of the 19th century, where it was just routine. The children, most notably, there'd be a large number of children that would die from this. That plateau isn't shown on this graph because very shortly after the federal ministry started maintaining these statistics, there was a vaccine introduced. And then it kind of, rather over a period of about a decade, it drops off until you get down into a pre-war era, and it kind of plateaus in there. And then it suddenly drops off after World War II. And the reason was a public policy change in the late 1940s that made immunization part of the normal social routine. There actually became that is to say, it's programmatic. It wasn't just that so the vaccine became available to physicians in 1926, in Canada at least. And then in 1948, it became programmatic, so that it was something that was part of the routine, part of going to a doctor, is that at certain ages, people would get these shots and the booster shots. So in the pre-show, I kind of referred to my part as the least interesting and the most important segment of this episode, and that's because I don't get to show the gruesome photos of ulcers and holes in people and the big plaques in the throat and the children choking to death and that sort of thing. But this is the part that there's public engagement, that you need to realize that the public has a role in health policy formation and encouraging your government to look at good evidence and to engage in evidence-based policy and practice. when it comes to around vaccine policy. So yeah, it drops off here. So we'll go down to this little inset graph, and you see there's a little bit of a bump in the 70s, but then by the 80s, it became pretty much required in all the provinces, and boom, it drops off to nothing. I should clarify that around the time that I had started getting involved in communicable disease research in human populations, we had gotten word of these are the world. So this case is the worldwide statistics, only starting after 1980. And again, it was pretty low, and there was a bump. We'd gotten word in the I got to do that yelling again. Just 1 second. All right, and they're in a different room of the house, which is awesome. So we'd gotten word of an outbreak in the what is now the former Soviet bloc countries, and not an outbreak. I'm sorry, that's the wrong term for it. But they were kind of losing control of diphtheria. They had pretty much eradicated it as well, but it started getting out of hand over there. They were starting to have a lot of cases, and this happens in the health policy world, where there's actually some surveillance. Obviously, each health policy jurisdiction surveils cases within their own jurisdiction, but there's some kind of looking over the shoulder, looking at your neighbor, peeking over the fence to see what's going on in neighboring countries because of these communicable diseases. Well, for obvious reasons, because these things are sort of dragged from one country to another. So there was a concern that there was going to be a worldwide outbreak through the late 90s. And if you look into the Russian data and the former Soviet Union data, what was happening? Well, first off, they were having some economic strife. There was some downshifting in social policy around things like vaccination, and there was even some health policy decisions made around eliminating booster shots, and then some kids just never got vaccinated. And as a result, there's a spike in the worldwide data. But if you were to tease this apart, you would see that this is due almost entirely to social political changes in a single portion of the world. And this is what happens when people turn their back on vaccination. You get a huge spike in one part of the world that is big enough that can actually bump the whole world's data.
Question for you, just to
yes,
go
please.
back to that previous one.
Worldwide, yeah.
98, post 98, 2000 period, it goes up a little bit. Do you think that comes down to the Wakefield thing where people got paranoid about giving their kids vaccinations?
I don't. I'm going to say no, because the initial outcry about the Wakefield thing was MMR. So as most people know, when you go to, especially for children, get vaccinations, they're in packages, and the name of the vaccine is always some random combination of letters that represent the set of diseases that would be inoculated against with that thing. So MMR referred to the Wakefield thing, which we won't go into because there's been a whole It's the Science Bitches episode on that topic. But this one is generally addressed by something called TDAP. I'm sorry, DTAP. The TDAP's for older. The DTAP. So now, granted, after that time, in some health jurisdictions, people just started being, instead of MMR causes autism, it turned into vaccines cause autism, and all use of vaccines start to go down, and you would see an increase there. I don't see that in either the worldwide numbers or the Canadian numbers, but people who've looked at the Western United States numbers would tell you a bit of a different story.
Okay, thanks.
Okay, so that's the worldwide ones. I wanted to say of the American situation. So here's the diphtheria cases in the U.S. Again, so their surveillance starts shortly before World War II, and again, they show the same thing. Once the Americans programatized vaccines, they have that same situation where you look at their diphtheria cases. And these are reported cases. Raw numbers are reported cases. These aren't the deaths from it. These are reported cases of infection, and the American population more than doubles in this time interval on the x-axis of this graph. But it's by the 1970s, it gets to be so low you can't even see bumps on the graph anymore. So the Americans had a very similar experience with it, and now, if you're to look at graphs that are sort of post 2010, they don't show on a scale like this. You're not going to see 2,000 reported cases, 10,000 reported cases. You're not going to see 1,000 reported cases. In fact, I thought I had the thing here on, look, we get the same pictures. Gruesome. I thought I had one open here on, oh no, it was this one. Okay, so now I've got to do the atrocious thing here. I've got to stop screen sharing, switch my screen share to the other window, which I think is this one.
This is what happens when people aren't as prepared as they should be.
That's true. Yes, I should have put way more time into this. This is the Health Canada notices on it, and literally their position on recent notices for diphtheria is none in effect. There are no notices going out either to the Canadian citizenry or to health practitioners that there's a concern over diphtheria. And then there's just this sort of standard information in here. So I wanted to touch briefly too on what happens when a modern clinician encounters a case of a disease that he could not possibly have received firsthand education because it's been eradicated in his healthcare jurisdiction. So we did have a case here in Edmonton, and I can tell you, having spoken with not the clinicians who dealt with that, with the ministry officials who reviewed the reports on it, apparently it was picked up pretty quickly. There are enough of these gruesome photos that get shown to students. Oh, I'm on the wrong one with the gruesome photos. There are enough of these gruesome photos that get shown to students, and it is on the board exams, that it was definitely caught fairly quickly. Plus, they understood the likelihood of there being communicability to other people that this was a public school child, had encountered, and the law in that particular province where this one case had happened permits forcible quarantine of people. Of course, everybody was quite happy to accept the government's instruction that they were to be quarantined and they were treated. And we now, according to the ministry, we now have 0 cases, not just in the province, but in the entire country. So what I'm saying is the eradication of diphtheria is attainable and maintainable as long as you have a compliant population that understand the wisdom of having inoculations and seeking treatment when they get the bacterial infection. And it's a funny time in the world, because when I say compliant population, there's a subset of people who hear sheeple, right? If you have people that are thoughtful and educated about the nature of these diseases, as hopefully their audiences sort of increase their thoughtfulness and their education listening to this episode, you can save yourself a lot of trouble, save your whole population a lot of trouble. But you have some people that when they hear compliant, they sort of hear sheeple, like do what the government tells you. If you understand it, you should be quite happy to do it. Let me say let me check my notes here on the adverse reactions. So why would you not want to get I thought by now we'd talk about the DTaP and the TDAP.
Yeah, I mentioned them generally with the sort of cursory list of the popular, unpopular components. So aluminum hydroxide, thimerosal, and some neomycin, some erythromycin, possibly formaldehyde, and so on.
Okay, I do recall that, and let's talk about. I mean, none of those things actually. They're things that frighten people a bit, but they generally don't lend themselves to adverse reactions. So let's talk about what happens when you or your child actually receives a TDAP or a DTaP, and I can say it had so amongst the adverse reactions, it actually has a fairly high immunogenesis rate, and this is about 50% of people. Well, when I say, I mean, everybody has an immunogenic reaction, but I mean, in terms of the a symptomatic reaction, about 50% of the people that get this combination, either the TDAP or the DTaP, they do report some mentionable symptom. It might be a very mild fever, pain at the injection site, kind of a feeling of malaise for a day or so. Just the sort of like, oh, my immune system seems to be fighting something. And that's exactly what's going on. Your immune system is reacting. Your body's kind of in a state of alarm over the 2 or 3 or 4 products that were injected into your body, the antigens to spur on the antibody response. Your body is actually reacting to it, and you do feel a little bit sick from it. And it's a pretty high rate. It's about 50%, but it's a pretty mild thing, and it goes away in about a day. And at the end of that process, your immune system is stronger. This is a point, I should note, I'm in the one of the Google Plus communities, the Society to Counter Anti-Vaxxer Nonsense. If you're here, leave because of Society to Counter Anti-Vaxxer Nonsense, post a howdy message in the live chat to let people know that that's where you're from. It's a great community, good discussion there, very, very sensible, caring people. And one of the discussions that I hope that people are getting into is about the idea about having a strong immune system, about strengthening your immune system, about weakening your immune system. There's this idea floating around that you shouldn't take vaccinations because it weakens your immune system, right? The thinking is a lot like you shouldn't drive around on a rascal all the time because it weakens your legs. You're better off to get up and walk around yourself and strengthen your legs. Right? They're saying you don't want to use vaccines because the vaccines weaken your immune system. You're better off getting the diseases and strengthening your immune system by letting it work on the fight. And it's crazy, crazy backwards thinking, because everything we've described in here about injecting these antigens, the things that don't make you sick, but they do encourage the antibody response. These vaccines, the injection of these antigens, including the MMR, including the Tdap and the DTaP, these combination vaccines actually are the things that trigger your immune system. They put your immune system to work. You might feel sick for a couple hours, 12 hours. You might feel a little bit of malaise for a whole day. But at the end of it, your immune system is actually much stronger. And you don't need to do so at the expense of encountering actual diphtheria in your body or the diphtheria toxin. It is very much for strengthening your immune system.
May I comment to reiterate? My experience of anti-vaxxers is very similar to my experience with creationists. They have a dogma that's in large part emotionally based. And none of the evidence that they cite to support their position, in fact, supports their position. And the data that Hogtie showed demonstrates that beautifully. In addition to that, we can do things like serology on patients to actually measure the degree of antibody response that's present in their blood when stimulated. So again, this is a thing that's really well understood. And to reiterate, the arguments against vaccination are not data-driven. They are, in many instances, understandable, but emotion-driven. And looking at the numbers, I think there is always a risk when you're administering anything that your body might react to. But the relative risks are vanishingly small numbers against diseases that can kill 1 in 10 people, for example, or maim others in hideous ways, measles in particular, rubella. Many of these things will either kill you or mess you up for the rest of your life. The odds of an adverse vaccine reaction that's significant is so small relative to the risks of having the disease that there is no credible argument against the administration of well-tested, well-understood vaccines.
But it is exactly right, and I will say a beautiful bridge into what was going to be my last point. I think as we cultivate and harvest questions from the live chat, I do want to take a look at some of the other data, which would be these anecdotal reports, where people are just actively discouraging each other from the vaccine, and Ian's point exactly pipes into that. The one teeny tiny correction I want to make is when you say the low risk of adverse reaction. Adverse reaction is a clinical term, refers to anything that was sort of an unpleasant experience, and it actually has a fairly high adverse reaction rate, where you feel icky for a couple hours or a day maybe. Adverse event are the things that very, very low adverse event. As a matter of fact, for the actual diphtheria toxin, this actually has a 0 adverse reaction event. There's literally no history of somebody getting the diphtheria toxoid to generate this antigenic response that has led to an adverse event. There are people reporting adverse reactions, this mild malaise, but there's literally 0 adverse events. It is often bundled. So when you see the Tdap or DTaP, the D is obviously diphtheria. The T is for tetanus. not to venture too far into the tetanus thing, but it also produces, it's also a toxin, and there's a toxoid. So just import the things we're just talking about with diphtheria to this is how we also deal with tetanus. There are some reported cases where the tetanus toxoid in a very, very small number of people actually does produce adverse event. They're very, very rare, and there's some thinking that the packaging into Tdap or DTaP is what's causing the problem. And there's some discussion about but the thing is, there are many, many diphtheria toxoids that are available that don't include the tetanus toxoid.
Yep. And there are some development products in which very specific antigens, so not the entire protein, are likely to be administered in combination to generate a much more specific response. But those are not in the clinic or even close to it at this time.
Yeah, so it's only going to get better. I mean, this is the way these things proceed. I mean, it's kind of a happy story the way vaccination has played out, and of course, they don't just get to a point. There's no finish line when it comes to vaccine research. They don't just say, oh well, we've got one now, so we're going to turn it loose on the population and then not look at it again. There's ongoing surveillance. There's ongoing development. It's going to improve.
I mean, the mantra that I have on this when I'm teaching is, therapeutic index is an interesting thing and a concept when you're treating people who are sick, but we're injecting or we're designing things to be injected into healthy children. So the best possible vaccine of yesterday is the minimum standard for any additional product. So it's a bootstrapping effect. But for the case of the diphtheria vaccine, it's a bit like inventing a better mousetrap. Because with today's technology and sufficient surveillance, this is a disease, as Hogtie said, that can be functionally eradicated.
Yeah, and I'll just respond to one comment here in the side chat, and again, as we harvest more questions and comments from the live chat, I'll get on to what is my last point here. There's some discussion in the internal chat here about the vaccine court. I just want to, and we can talk about that in a second when I'm, just let me finish my last point here. I do want to say that when I'm explaining this, when I make my point about that the Canadian position is that there are no adverse events attributable to well, any number of vaccines. I mean, today we're talking about diphtheria. I do want to make it clear, Jade, for your benefit, that we take no guidance from the vaccine court in terms of when we're trying to tally what the actual events are. We look for things that are objectively attributable to a particular cause. Can I let that answer be adequate before you go into explain the vaccine court? Because I don't want to have it derail my point. But I'm saying somebody can go ahead and say that they've heard of bad stories, but that's not to take away from my point that when I say that the Canadian governments take this position, that's based on analyzing data that would not include vaccine court judgments, because they're not binding on our opinions about, okay, whatever. Let me just go into my last point for
that. That's fine. Yeah, and they're also no fault. So yeah, go ahead.
Okay, and we are hearing a bit of crackle from you, Mike. So I wonder if you want to F5 while I go into my last point. And okay, okay. So, but it is worth mentioning. Let's not bury this. So we do have organizations like Vaccine Choice Canada that do have listings here on adverse reaction to the Tdap vaccine. This is not specifically about diphtheria, but most Canadians who are going to encounter vaccination for diphtheria are going to do so in a combination, whether it's the Tdap or the DTaP or the what's the other one, the PD or DP or TP, whichever the third one is that uses the booster. There's a story here right on the opening page about somebody who, my knowledge of the true risks and harm vaccinations can cause comes firsthand. Right, and then there is a firsthand story that goes on for paragraphs here. This person went through a terrible ordeal. This person is obviously quite convinced that it was attributable to the vaccine, the combination vaccine. That is, there's a Think Twice is an American organization that reports to be the Global Vaccine Institute. They have many stories here. You see this Q&A. My son at 9 months was given DTaP vaccine 3 days ago. He was very tired after the shot, but otherwise fine. And then started to have a high temperature and no appetite. And then he's going jerking while he sleeps. My daughter, 5 years old, was given this shot because of school insistence. So this parent objected to it and then started having a severe local reaction, which is pretty common. But then after a time, she has autistic tendencies, right?
Yeah.
So.
Hogtie, can I mention another organization, because for our American viewers. The National Vaccine Information Center has a very polished website and looks like a government organization, but it's anti-vax wingnut central. Let's just be aware.
Could you repeat the name of that, Ian?
National Vaccine Information Center.
That's a weird thing that happens in the US, I notice quite a bit. They'll make something the national this or the federal that or whatever, but it's not like they're not actually run by the United States government. I mean, not to, just because something comes from the United States government, I wouldn't say means that it's it's credible, but at least there's some oversight there. But it seems a lot of these organizations call themselves the American this or the American National Organization of that, and they're actually just random, private bodies that do this.
I wonder how much of it is because no one seems to have another one the same, so we might as well call it a national one because we're the only one so far.
Yeah.
I've posted the link in the chat, but they describe themselves as a non-profit organization dedicated to prevention of vaccine injuries and deaths through public information, blah, 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 blah. And it's sort of science-polished woo and anecdotes. So that was the point, just another.
Yeah, and if I could jump in real quick. I mean, I understand that parents are going to be worried when kids get injections, and whether that stems from needle phobias or experiences they've had in the past or they've seen. We have to face the facts that these vaccines contain a minimal amount of antigens, things that will cause the patient's immune system to react. We're talking at most 20 antigens in a series of vaccines someone would get in the clinic. Whereas when your child's out playing in the yard, I don't think most parents bat an eyelash if the kid scrapes their knee and they get a little dirt in it. In that instance, that kid has 1,000+ antigens directly into the bloodstream. It doesn't make any sense why they place such a high level of importance and stress that, well, the antigens that come from the vaccine are so much worse than the antigens that we get out of nature, when we're just trying to mimic the ones in nature and target the ones that have the greatest benefit.
And I find it fascinating because I've spent a good part of the last 10 years working out ways to both minimize what we would call process or product related contaminants and to measure very accurately the tiny amounts. We're talking about parts per million of the things people freak out about, staggeringly getting confused always between toxicity and dose and how the two are related. So again, it's an emotional argument that really cannot be backed up by data.
It can only be backed up by misinterpreting the data with possible maliciousness.
Sure. And that's the only kind of comment that actually hurts, when people say, well, vaccines are designed to kill or sterilize and reduce the population, and I've got to be evil for being involved, when in fact the opposite is exactly the truth.
There is certainly an extreme wing to the anti-vax movement that are impenetrable to evidence or to good reasoning. And they really, in my view, can't be the target audience for this sort of a message. I mean, the thing that sort of breaks my heart about it is that there are so many people, and there are people in my family in rural communities that are seeing a lot of the anti-vax stuff, and they kind of think about, well, they put a lot of stuff in vaccines, and they repeat a lot of these tropes about how you really want to strengthen your immune system, so you're better off not to have vaccines. I think that group of people is a better recipient for a targeted message about how vaccines is, you're putting antigens into your body to strengthen your immune system. Just like when your kids go outside and play. That's a great point. When your kids go outside and play and they fall down and scrape their knee, they get all sorts of antigens put into their bloodstream, and their body reacts to it, and that strengthens their immune system. And when a doctor or a nurse, or now is legal in Canada, a pharmacist injects antigens into your bloodstream, your child's bloodstream, those are very carefully produced, safe antigens that are going to give your kid protection against very, very dangerous things out there in the world. And
Yep.
they do so by strengthening your immune system and your child's immune system. This is the message that's got to get out there to that group of people, for which there could be some penetration, not the absolute nutters that think that we're implanting microchips with the MMR and stuff like that. But maybe I'll kind of wrap up on that point, because I think people get the last thing I was screen sharing here is you can go to any number of sites on the internet that do have these stories about, when I had this, I had to, 5 minutes later, the paramedics had to pick up my daughter. And these are very, very sad things. These are very difficult things. And, I'm a parent that's had the horrible thing happen to my kids, where you've had to get an ambulance, or you had to take them to the hospital, and they're in a hospital for 4 days and you're sleeping in the hospital. I'm just going to scroll down as I completely identify with these people. I know how painful it is. But the problem is, parents go through this stuff, and if I guess let me say it this way: if it were the case that the DTaP actually caused some health problem in 1 in 100,000 children, then we would know it, because we surveil these things very carefully, and we would have a strategy to deal with it. We must. It's the whole Hippocratic oath. You could do no harm. And if it were the case that the actual doing of this vaccination, the actual medical intervention, is the thing that causes some sort of problem in 1 in 100,000 kids, the system would necessarily have to react to that. Physicians couldn't tolerate that. They would have to be prepared. There would have to be a system to detect those 1 in 100,000 and to deal with those cases. And even if they couldn't, it would still be better than the plague that happened to children in the late 19th century and the early 20th century.
Absolutely.
Even if that were the case. But it's not even the case. So again, I've gone through the data. There's nothing indicating that the DTaP or the Tdap or the 2 antigen form, that these things are not causing these adverse events, that these stories are out there, and people need to know. You need to live in a country, you need to encourage good health policy that make it so that when your kid is sick, you have a place that you can go to and have your kid treated. These things are not attributable to vaccines, and part of the way that we're going to be able to prevent these kind of plagues, like what used to happen 100 years ago, is to have good education around vaccination, and to all strengthen our immune systems and strengthen the immune systems of our children with properly scheduled vaccinations.
Absolutely. And to comment very briefly on the vaccine itself, every single dose of a vaccine is traceable back to its batch and to the primary data around its manufacture. And to be released to the clinic after extensive testing for a number of years at minimum, about 20 different variables have to be within very closely defined specifications, tested using methods that are fully validated so that we know how accurate they are, how specific, their range. Everything has to be fully regulated so that we know very much what's in every single dose. This is a highly, highly regulated industry, and the quality of the products, the quality control on the products is really remarkable.
Yeah, Ian, I want to add to this whole vaccine conversation. A lot of the times you'll hear people who are on the anti-vaxxer side say that, well, these pharmaceutical companies, they're just in it for the money. They want to shell out as many vaccines as they can to make the biggest profit. And that story just doesn't ring true to me. If you look at the profit margins, vaccines actually have the lowest profit margins of pretty much almost any medical device.
Yeah.
And that goes, let's talk about Band-Aids. They make more money selling Band-Aids than they do selling these vaccines. And where the real money is are medicines that these patients have to take every day. You take a vaccine once every 10 years. There's just not a good profit strategy in that. So that argument falls completely flat.
I remember looking into this and figuring out that the cost of not vaccinating people and the illness that would, propagate as a result of that, they would make more money from loads of sick people than they would from, by not vaccinating people than they would from vaccinating people. It is in their least best interest to vaccinate people.
Absolutely. One treatment case There's any number of diseases you could do this analysis for, and it certainly would fit for diphtheria. But one treatment case is way more expensive, and even specifically for the what do you call it, the drug producers, than literally thousands of vaccination cases. And keep in mind, Cliff, I'm not sure. I think that you guys do this over there. Here in Canada, remember, we don't have one healthcare system, right? Each province has its own healthcare system. But in my province, in each individual province, they actually bulk negotiate their vaccine purchases. So whatever you think a vaccine costs, if you think it's $5 a dose, my government calls up the manufacturers and says, "We'll give you $1.50."
Yeah.
But we're ordering 1.3 million.
That's
Like literally, we are totally like, we're getting bargain basement prices from these vaccine manufacturers because we order so many of them, and they're making money, but only because they prevent literally millions of cases of, or they vaccinate millions of people in the process. Not a big profit margin.
The money is, as Hogtie says quite correctly, in wholesale, but increasingly the research wings of vaccine companies are borrowing vaccine function to generate treatments for some tumor types by stimulating an immune response against self but cancerous cells. So it's an expanding field, and the boundaries between vaccine development and biopharmaceutical development are becoming more blurred over the last several years.
Could I just tie all this together, please?
Yeah,
because we've talked about the mechanism of action of the toxins, and we showed some icky photographs of the actual local infection, but we didn't really discuss what the overall effect of that toxin is. We talked about cell death, and that's it. So what does that actually mean? Well, cell death, we call it necrosis. An area of cells dies. And with diphtheria, the most common areas for this necrosis are the heart and the liver. So that means a large portion of the heart muscle dies, or large portions of the liver die off. That kills the patient. It's not a nice way to go. For anyone who's interested, you can go to Wiki and look up diphtheria and either heart or liver necrosis. And you can see some pictures there of what that toxin does to those organs, and compare that to the raised temperature that a vaccinated child gets when they get the vaccine. And ask yourself this question: which would I prefer? Because you see the difference. I'm going to bring up smallpox here for a very good reason. We eradicated the smallpox virus, and by doing that, we eradicated the disease. We were able to do that because there is no animal reservoir for the smallpox virus. And it can't exist out in the environment, out with a host. So we were able to completely eradicate both the virus and the disease. With Corynebacterium diphtheriae, it's a really robust little chap. It can survive in the environment, in soil, fomites, all the rest of it. Because it's not impervious completely, but it's really strong. We can't eradicate the bacterium, but we can, with vaccine, eradicate the disease. And in fact, I would say that given the fact that we cannot eradicate the bacterium, then vaccination is all the more important. I just wanted to throw that in there because we hadn't really tied in the end result of the action of the toxin. And also one of the problems with these robust bacteria.
It might be slightly worse than that too, Fiona, because and you do mention the mechanism of death. So it attacks the heart, and you get the myocardial necrosis. So there's actually large sections of the heart tissue will die, and that often leads to the death, especially in children. Infants, it's really bad in infants. Perhaps arguably worse than that is that some of those patients survive. Now again, this isn't something you see in the modern era because the antitoxin has been around for a very long time, but there are case reports from the last part of the 19th century. I think maybe the reason why we skipped over this, we're trying to keep the time short, and we didn't really want to sort of venture too far back in history. But there are case reports of children that had survived with from like say the late part of the 19th century, the early part of the 20th century, before the antitoxin was available, and they survived the necrosis of the heart tissue, and of course they lived their lives with a partial heart or partial liver, significant brain damage, respiratory problems that were persistent through life, and it's pretty gruesome. I mean, it's not something we should try to frighten people about in 2018, because you'd have to be living in pretty bizarre circumstances to not get the antitoxin. Or I mean, I shouldn't say that, because I mean, if you're a religious nut living in Calgary, right, like what happened a few years ago with that appendicitis patient, right? Maybe somebody wouldn't get the antitoxin because they were praying instead of taking their child to a doctor. But yeah, my point is there are some pretty gruesome case reports from the early 20th century before the antitoxin was used of people living with partial hearts afterwards.
Yeah, I was going to say, right, Jade, I'm sorry, you've been so patient, dear. Go for it, hen.
Darn it, I did that thing where I unmuted myself and then immediately muted myself. So I'm here. Well, I've been patient. We have people here on this panel today who knows so much more about this than I do. I'm more than happy to just listen. So they're basically like Cliff brought up something that I'm going to bring up right now before I go into what I was learning, as the damage that's caused to people by various diseases. And we are in an era where people aren't seeing the kind of diseases that were around when I was a child. When I was a small child, there was a polio epidemic in this country. And my mother was afraid to let me go out and play with other kids because of the polio epidemic. I grew up around people who had been damaged by polio. I myself got the measles, and it was so bad that it has caused me a permanent hearing damage. And that's because there was no measles vaccine back then. But people aren't seeing all these cases of the measles and the mumps and chicken pox and and diphtheria and tetanus. So people aren't seeing these things, so they don't think they're that scary anymore. And that contributes to the reason why some people don't want to have vaccines. Where are these diseases? And it doesn't take long. We've talked about measles before. I'm not going to go into it here, but the measles plaques can last for a very long time. There was a case where a baby got the measles. This was in Southern California. A baby got the measles because the baby's older sibling played on a team that was coached by a man who had the measles. Mm-hmm. So the measles plaque was undoubtedly carried home by the sibling, and the baby was infected with the measles. So, but we just don't see these things anymore the way we used to. And so that's why people really are unaware of how dangerous it is. The so-called childhood diseases are not, are dangerous. Just because a lot of people aren't injured by them, there are people like me who have permanent damage because of this. And as Cliff pointed out here, he says, how much does it cost me for my hearing aids for a lifetime? Because of the measles and because my immune system was compromised and I had ear infection after ear infection after ear infection. So that's it's a scary thing. We have to have children vaccinated. I don't ever want a child to go through the horror I went through, and I do remember it. The other thing I wanted to bring up was the additives that are in vaccines. If people are frightened by them, you can look them up. The CDC has them listed. You can look up what additives are in vaccines, and you can look up why they're there. But I wanted to bring up three briefly that there's a lot of fuss about. But I also wanted to mention something about toxicity. People are so afraid of things that are toxic. Caffeine is toxic, at certain levels. And there are people who have died from having too much caffeine. Now, usually they have another problem as well, which is what the caffeine contribute. So the caffeine was a contributing factor to their deaths, not the only reason for their death. But so, yes, caffeine, we get caffeine all the time, so it's dangerous. So when you're talking about something that's toxic or dangerous, you're talking about the amount of it. It takes a lot of caffeine, for one thing, for it to be fatal for a human being. But the amounts that are these additives in the vaccines are very, very tiny.
And
But
very, and extremely well measured as well.
Right.
And you make an excellent point, Jade. Can I just say, if anyone has any specific questions about the compounds that are present in any of the vaccines, please send them to me.
Right. And I wanted to mention, so I want to mention three things. First of all, I wanted to mention mercury, which is always brought up in, you know what? I always mispronounce this. Thimerosal.
Yep, that's
It's one of those words. Thimerosal. Okay. It's one of these words that I keep, with me and my dyslexia, I keep, sometimes with words, I either leave out letters or add them in, those thimerosal, which is a form of ethyl mercury. Now, it's methyl mercury is deadly. It's very, very dangerous. I know some people probably remember Japan and the contaminated tuna and that whole thing. That was methyl mercury. Some people don't understand the difference between methyl and ethyl. Without going into the chemistry, let me point out that we drink ethyl alcohol. Methyl alcohol will kill us or make us blind. It's also known as wood alcohol. So the type of a compound you're using is very important. Other thing I wanted to point out is I put the paper up above Cliff, and you can put that in the outside chat for people. So they did a study of babies. The babies were given vaccines with ethyl mercury in it, right? And what they discovered is that the substance basically just flushed right out of their bodies. So they're not retaining it. The children are not retaining this ethyl mercury substance. And if they aren't retaining it, it can't hurt them. Okay?
Can I?
The second thing I wanted to bring up.
Comment briefly?
You want to say something, Ian?
Yeah.
Sure.
Again, it's a fantastic point, and it's great to hear the fine-grained chemistry. Again, this wasn't a guess when this type of product was started to be used in people. The pharmacokinetics of each of the components of the vaccine are studied during the animal trials, which are a legal requirement before any kind of human clinical interaction. So it was well understood that if you give thimerosal at doses relative to body weight, as is the case for the vaccines, you pee it out in a matter of hours. That's it, Jane.
So the other thing I wanted to bring in, and I'm dealing with Felis interruptus at the moment because Freddie has to get into everything. Go, Freddie. Is formaldehyde. Now, a lot of people don't realize this, but our bodies produce formaldehyde. So formaldehyde isn't such a very, very scary thing. For example, when we break it, sometimes when we get methanol in like citrus fruits and juices or fermented beverages, our bodies break it down into formaldehyde and other products.
That's right.
And so, our bodies produce formaldehyde as a result of DNA demethylation. And our bodies will break formaldehyde that we ingest down very quickly into formic acid. So we make formaldehyde. It's not such a scary thing. And there's probably more formaldehyde in the pear you will eat than in any vaccine you'll take.
That would be, I think, orders of magnitude conservatively more, because the glutaraldehyde and formaldehyde content is down at the low parts per million. And very often it's impossible to quantify at such low levels. So the threshold is marked at the lowest possible sensitivity of the method. So in a vaccine dose, we're talking about nanograms. That's 1 × 10⁻⁹ grams.
Yeah. And there's a lot more than that in pears and other fruits and other substances we ingest, because all life on this planet is related. We all have similar chemical processes, and producing formaldehyde is one of them. The last thing I want to bring up is aluminum, and this terrible fear of aluminum. So I've been doing research on different elements for some videos I'm going to be doing on chemistry, and I'm quite literally going through and looking up every single element and finding out its sources, what it's good for. One of the things I found out about aluminum is it's everywhere.
Yep.
Because our modern world uses aluminum so much, aluminum dust is everywhere. You are inhaling aluminum dust. Aluminum dust is on your body, just from existence. So if you're going to be frightened by aluminum, you'd probably better, I don't know, go out in the woods somewhere. I'm not even sure that would work because it's probably going to rain sometime, and the rain is probably caused from evaporation from a place where there is aluminum dust. So I'm not even sure that's going to work. But aluminum is everywhere.
There's another subtle point there as well. The aluminum in vaccines is almost invariably aluminum hydroxide salt, which is a very different thing from the heavy metal.
Right.
And people get confused between them. And the analogy I use is, well, sodium is a metal and chlorine is a toxin. And separately, both of those things will kill you. Together, you put them on your chips.
Yeah, we do. Sodium chloride is a marvelous flavor enhancer. So I want to mention those three because they're brought up a lot. And if people are concerned about these, you can look them up. You can ask us questions, and someone here will probably know the question: why is this particular thing in a vaccine? And there are some very good reasons for why certain additives are in vaccines, like fungicides. We don't want fungus growing in our vaccines. But the last thing I wanted to bring up is the vaccine court. The vaccine court here in the United States is very misunderstood. I have to look this up for a second because I always forget the actual name of the thing. We call it the vaccine court, but it's actually the National Vaccine Injury Compensation Program, or VICP. Now, the way it came about is that during the 80s, a lot of people were suing for everything. It was a time, for those of us who were there might remember, people were suing everybody for everything. It was like a national sport. And the vaccine companies were being sued for alleged injury to vaccines. The fear was that these lawsuits, and unfortunately juries tend to sometimes, the poor kid is hurt, we'll give him a lot of money. Vaccine companies, these pharmaceutical companies, they've got a lot of money, we can give them some. So it's not always as rationally based as you would like. So the fear was that these big compensations given to alleged injuries due to vaccines were going to cause the companies to get out of the vaccine business. And this is extremely dangerous. I've pointed out before, I would really never, ever want to see a child go through what I went through with the measles, nor would I want them to go through what I know other people went through with polio. Of course, we've just about eradicated polio, but there are other nasty diseases out there. So our government, go ahead.
As a brief thought experiment, imagine the impact of the anti-vaccine lobby being successful and all of the childhood vaccines being removed from the schedule. What would happen in 2, 5, 10, 20 years? There'd be a whole pile of dead kids. Now, would the same people be protesting at that? And I really hope they would. And thank goodness that is a thought experiment, at least in our parts of the world at this time. But these things do not always go away, especially with the animal reservoir for some diseases. It's going to be a constant thing that we have to be on top of. And the effects have been manifest over the last 100 years at least. And it's been one of the two major impacts in human health: vaccination, effective medicine, of course, but vaccination and good plumbing have saved more lives.
Clean drinking water.
Yeah.
So get to know a vaccine developer and a good plumber, and I recommend a mechanic too.
And the one thing I want to mention is it was the DTaP vaccine that people were suing over mostly. And several pharmaceutical companies did threaten to get out of the business. So this was scary stuff. So laws were passed to develop what is colloquially referred to as the vaccine court. Now, the point that people miss about the vaccine court is the vaccine court is a no-fault court. It does not determine whether a company was at fault or a vaccine was at fault. What it's looking at, was someone injured? Is it possible that the vaccine contributed to that injury? That's what it's doing. It's not claiming, the settlements in the vaccine court are not claiming that the vaccine actually caused the damage. It's saying there is a possibility. There are sometimes rare side effects, there are allergies, and what the vaccine court is there for is to give some people money to make their life easier because they have incurred some damage, which may or may not be related to the vaccines. There are rare reactions to vaccines. There are allergies to vaccines. So these things are a possibility. Now, there is a case recently that some anti-vaxxers have been touting where a child was given a huge settlement from the vaccine court. And they point to this as, aha, you see. The part that they miss, first of all, this child had, I don't remember the exact case, I didn't look it up. This child had a catastrophic reaction to something terrible happened. But what they also discovered is this child had a unknown mitochondrial situation.
Yeah.
And so the vaccine court did award the child a lot of money, awarded the child, the parents or the guardians of the child, millions of dollars to help care for this child throughout the child's lifetime because of the serious damage. It doesn't mean that the vaccine caused it. And even if the vaccine caused it, there was this underlying unknown condition. And there is no way at the present time to test everyone for everything before they get a vaccine or before they take a medication. There will sadly be terrible things that happen.
And
they're rare.
There are unpredictable things clinically that can happen, I'm sure. Can I comment again briefly?
Sure.
When I was working for a significant vaccine development sort of multinational company, we were drilled extensively on what happens when there is an adverse event, as in something really serious, or a problem with the vaccine. Does it look right? Anything like that. That will put an entire department into immediate action. And there are various options, but typically everyone errs on the side of caution. And what we would do is recall that entire batch. And, that's a massive logistical operation. It's extremely expensive, but there isn't a credible biopharma company who would not respond in that way, because putting your patients at risk is a really, really bad business practice. So if you want to see a rapid response from a global company, an adverse reaction of that type, an adverse event, so a severe clinical issue, will induce that response immediately. And there is a legally defined timeline for that, which I'm sure Hogtie is more familiar with than I am. I'm not quite in that business right now.
Now, the last thing I wanted to bring up is an interesting historical point, and I'm not going to take very long on this because you guys anyone who's interested can look this up. In 1924, and remember, the vaccine for diphtheria wasn't developed until 1926. In 1924, the community of Nome, Alaska was getting ready to shut down for the winter. And when they shut down for the winter, they shut down for the winter. Remember, 1924. No airplanes, no modern conveniences, none of that. In this town, there was one doctor and 4 nurses. Now, they'd run out of the diphtheria antiserum. Not vaccine. Remember, no vaccine yet. And the doctor was extremely concerned about it, and he put in an order for more. It didn't get there. A case occurred in the town. And this was very frightening because at this time, one case of diphtheria, there are going to be more. And so the doctor called for help. and help arrived. It took 5 days of people going by dog sled all the way across Alaska. 5 days, one person, one dog sled after another, carrying the antiserum to Nome, Alaska, in time to stop an outbreak of diphtheria.
Mhm.
And it's a fantastic story to read about. And it is the basis for the Iditarod sled race today. So I'll just leave it at that. But the thing is, at one time, people didn't quibble over, things that would stop a disease. These people were willing to stress themselves, to stress their dogs, because the only way to get there at that time was dog sled. Day after day, person after person, dog sled after dog sled, carrying this life-saving substance to this, at the time, much smaller, tinier community of Nome, Alaska, to save people's lives. And that's where I want to end it.
Thanks, Jade. Right. I think we've basically covered everything we needed to cover tonight. I want to... Oh, sorry.
Sorry, I think Hogtie has a question from the chat that maybe we could address.
Well, I was going to let it go in mind of the time, but maybe I can charge through it really quickly. Albeit had asked, why is it that you can use combination vaccines to inoculate somebody against a variety of diseases, 2, 3, 4, depending what's in the vaccine preparation. Maybe I can give you sort of a quick, and I'll package it with a little bit of vaccine advice too. And basically, you don't want to get vaccinated when you're already sick. If you have a cold or you have the flu, take a week off, go back the week afterwards. Not that it's dangerous to get vaccinated when you have an illness, but there's some evidence that it actually makes your illness a little bit worse, a little bit harder to recover from it. Not that there's any problem, but you'll be fine. So let's say you go into the person who's generally healthy. These vaccines carry a very small number of molecules of each of these different antigens. And Fiona was suggesting we actually do a hangout on the immune system. I think that'd be fascinating. I think maybe I can just tease people with this idea that there's this great video by the Kurzgesagt channel, now called In a Nutshell, because nobody knows how to pronounce the actual Kurzgesagt channel name. The Immune System Explained 1, Bacterial Infection. This is just such a neat little video that explains the different cells that are in there. People should watch that. It's such a tidy, neat little explanation. But the thing to remember is that in there they explain about these accessory cells, the dendritic cells that are able to, they're sort of the not policemen, but the detectives that go along and say, oh, what's this bit of foreign material? What is this? And they kind of analyze it. And then they from that, they sort of trigger these responses. The thing to remember is that those cells are available by the trillions. So you can imagine a city where there's a crime going on here and there's, a speed trap over there. You can take something like the Tdap or the DTaP or the MMR, and there'll be enough dendritic cells out there that some of them will be able to take a look at the 4 compounds. Some of them will be looking at one compound from one vaccine, another one will be looking at some other compound from that vaccine. So there's certainly no harm in taking multiple vaccines. And in parallel, your immune system has enough resources that it's able to actually produce immunity, a full inoculation against a whole variety of things. But again, it's a little better that you go in when you're not already fighting a cold.
One vaccine available is a pneumococcal vaccine for pneumonia, is caused by pneumococcus, that has 24 variants of polysaccharide, the outer skin of the bugs, to prevent multiple different strains. And they're very similar, subtly different. And this just gives you a response that will basically deal with anything of that type. It's one of few cases where you can take all of that and put it together in one product. But another one is the HPV vaccine, which has, I think, 3 variants present in it at present, but that will be adapted based on the molecular surveillance of the disease as it happens. So these things are adaptable and constantly developing in response to the clinical environment. So it's a very exciting field. The flu vaccine is trivalent this year as well. So 3 different variants covered in that. And increasingly, the advances in vaccine development technology enable reactive vaccine development, for instance, in response to a flu epidemic.
Okay. So as I say, I think that, have we covered everything? I'm just picking up Hogtie charts.
No, there's a lot more to cover. Like, what are dendritic cells, Hogtie?
No. Next week, schistosomiasis.
I have an immunology joke, and this is a true story. I was flying into Allentown in West Virginia to talk to some people at the medical school there. And I arrive in this place, kind of dazed after multiple flights. And the customs guy, when I arrive in the US, goes, "Who are you and what are you doing here?" I said, "Well, I'm an immunologist. I'm going to talk to some people." And he goes, "Really? What are Peyer's patches?" And I'm like, "Well, that's a very interesting question." And I told him these are areas in the gut that sample the food antigens so your body doesn't respond to them, as in selective immunosuppression. He says, "That's very good. What's the equivalent organ in a chicken?" This is 2 a.m. in an airport. And I'm like, "Are you serious? You're not going to let me into the country because my chicken immunology isn't up to snuff." But actually, I know the answer, and it's the buccal pouch. And how do you know this? He says, "I've been in this business for 40 years, son. You pick up things." So this guy I was amazed because, wow, what a new question. The first part,
That is amazing.
The first part, that's impressive. The second just had me on my back. Like, you can lock me up if you want to, because you are the master.
And I think on that note, it's a good one. It'd be very worrying. I wouldn't have been able to answer the chickens, I must admit. But then again, I've not had a lot to do with chicken anatomy, pathology, anything, because I'm not into chickens.
I was rolling on the floor because this guy,
surprised.
the cop mustache, the whole thing. It's like, welcome to America. What a wonderful place.
You didn't expect I don't know, immigration cop to use foul language.
Oh,
Nice. Nice.
Right, we're ending it on that. That's it. But listen, I want to thank the audience for sticking with us. I really, really hope that you people out there got something from this, and I hope you found it interesting. I would also hope that you keep an eye out for our next one. Now, one of the topics that we're going to cover is, what is the immune system and how does it work? And I am suggesting that maybe we should do that one next.
Yeah.
Just because we have been talking about the immune system quite a lot in the last couple of hangouts. So I think maybe we should actually cover what the immune system does, what's it for, how does it do it?
It's
to give people an idea, so that now when we say, and then the immune system does such and such, they've got a picture in their head of what's going on.
It's a beautiful story. It's fascinating. It's complex. It's super nifty, and it has the appearance of being designed, which does not mean that Jesus did.
Right. So thank you to everybody who's participated. Thank you to everybody who has watched. It is really appreciated, and I hope you found it helpful. And with that, say good night, everybody.
Good night, everybody.
Say good night, everybody.
Good night.
Night.